Here's something that should bother you. The most sensitive early cancer tests we're building right now, they don't look for what's broken. They measure how randomly broken things are becoming.
Which is, okay, that sounds like a weird distinction, but it's actually everything.
So, liquid biopsies. The idea is you take a blood draw and you detect tumor DNA floating around in the bloodstream. No scalpel, no tissue biopsy. It sounds almost too clean.
And for late-stage cancers, it kind of is that clean. Like monitoring treatment response, tracking resistance mutations, liquid biopsy is already standard practice. The problem is early detection.
Which is the whole game, right? Because if you catch it at stage 1, survival rates are dramatically better.
Dramatically. And here's where it falls apart. The most prominent test out there right now, the Grail Gallery test, it detects over 50 cancer types, 92% sensitivity at stage 4.
Which sounds amazing until
Until you look at stage 1, 18%. Across all cancer types, you're missing 80% of early stage cancers.
80%.
80%. And the reason is biological. Early tumors just don't shed much DNA into the blood. A small, slow-growing mass, it's barely whispering. The signal is drowned out.
So the question is, are we listening for the wrong thing?
Exactly.
So this is where the Johns Hopkins work comes in. And this is the part that genuinely reframed how I think about this.
Right. So, okay, traditional methylation-based liquid biopsy, and methylation is basically this chemical tag on your DNA that regulates which genes get switched on or off. Traditional approaches look for specific changes, like this region should be methylated and it's not. Very rule-based.
Find the broken rule.
Find the broken rule. But cancer isn't really a broken rule, it's more like a system losing its coherence. Cells are dividing fast and sloppily, and the methylation patterns across the genome are becoming chaotic. Not broken in one specific place, just increasingly random.
Right. So Johns Hopkins developed this thing called the Epigenetic Instability Index, the EII. And instead of asking, is this region methylated or not, it asks how variable is the methylation from cell to cell across the tumor.
Which is, that's a fundamentally different question. You're not measuring a signal, you're measuring the entropy of the signal.
And the numbers are remarkable. 81% sensitivity for stage 1A non-small cell lung cancer, 68% for early breast cancer, at 95% specificity.
Compare that to 18%.
Right. Same stage, completely different approach.
And they identified 269 specific CTG islands, these little genomic regions that are like the optimal windows to observe this stochastic variation. Random forest classifier on top of that, and it cuts right through the noise of normal aging.
Okay, but hold on. How do you even sequence this? Because traditional short-read sequencing, you're fragmenting the DNA, you're doing bisulfite conversion, which chemically degrades it.
Yeah, this is where nanopore sequencing becomes critical. Oxford Nanopore literally pulls a single DNA strand through a tiny protein pore, measures the electrical disruption as bases pass through, and it can read native modifications, methylation included. No bisulfite conversion, no PCR amplification.
So you're reading the epigenome directly.
You're reading the epigenome and the genome simultaneously from the same molecule in real time. And it can do this at really shallow coverage, like 0.05 times, which makes it economically viable for actual screening.
Which matters a lot. Okay, and then the computational piece. Because now you have this incredibly high-dimensional signal. Thousands of methylation sites, variation patterns across the genome.
And this is where quantum kernels enter the picture. And I want to be careful here because there's a lot of hype.
Understatement.
But the specific problem they're suited for is real. Classical machine learning, when you have thousands of features in a small cohort, rare cancer subtypes, you can't get a million patients, you hit what's called the curse of dimensionality. The search space is just overwhelming.
So, what quantum kernels do is, and correct me if I'm oversimplifying, they encode the data into this exponentially large mathematical space called a Hilbert space. And the quantum hardware physically evaluates the similarity between data points in that space.
Yeah, the inner product. And the key thing is you get that high-dimensional feature mapping with relatively few parameters. UChicago did this with exosomes, these nanoscale vesicles that tumors shed, and they used a quantum algorithm to read the electrical fingerprints of the particles, zeta potential, conductance, electrophoretic mobility, and they distinguished cancer-derived exosomes from healthy ones with minimal training data.
Wait, really? Like minimal training data?
That's the headline, which for rare cancers or specific subtypes where you just can't build a massive annotated data set, that's potentially transformative.
Now, the barren plateau problem is real, though.
It's real. As you scale up qubits, the gradients in the loss landscape can vanish exponentially. The algorithm gets stuck on a flat surface with no sense of which direction to improve. And hardware noise creates what they call noise-induced barren plateaus on top of that.
Say it's not a wholesale replacement of classical ML.
No, it's the framing that makes sense is hybrid workflows. Classical computers handle the optimization, quantum processors handle the specific, intractable, high-dimensional kernel evaluations, surgical use.
So, zooming out, what does this actually look like as a clinical pipeline in, say, 10 years?
I think it looks like your routine complete blood count, which everyone gets fed continuously into a classical ML model tracking your immune trajectory. And when that trajectory starts drifting toward a pan-cancer vector, it flags an algorithm recommendation for a deeper test.
And that deeper test is nanopore-based methylation liquid biopsy, running the EII.
Measuring entropy, looking for chaos before there's a mass to find. And if the cohort is small or the subtype is rare, quantum kernels on the back end doing the classification.
The definition of early detection shifts from finding a small tumor to detecting the mathematical signature of cellular loss of control
before a traditional tumor microenvironment even forms. That's a genuinely different game.
And the Minon, Nanopore's portable device, is small, affordable-ish. This doesn't have to live in a centralized mega lab.
Rural clinics, under-resourced settings, the decentralization case is real.
We went from blood draws to entropy theory to quantum Hilbert spaces in under 6 minutes. So, hope that landed.
Should have brought a whiteboard.
I'm Nova.
And I'm Alex.
And this is Code and Cosmos.